can you unmute yourself and
Yes, I have.
okay.
Yeah.
Yeah,
Can you hear and see me now?
very good. Yes, I can see
Excellent.
it.
Sorry for that little glitch. Welcome to everyone who is listening in on this little talk where I'd like to share some of the insights through my own scientific journey. As Caroline has said, I trained originally as a physician treating cancer patients, and my journey then took me to spending over 25 years in Cambridge, primarily at the Medical Research Council's Cancer Unit, and will be bringing me to direct the Cancer Science Institute at the National University of Singapore in the next few days. So for those of you who have not previously used this system, please be aware that to ask questions during this webinar, you must use the Q&A button, which is in the bottom of your screen. After the talk, I will either invite you to unmute yourself, or one of the other panelists will actually respond to you directly via the Q&A function. I apologize if your question cannot be answered during the webinar, but I will be available at the CSI e-booth immediately afterwards for those of you who have further questions to ask. So I think many of you will be aware that all human diseases arise because of complex interactions between our environment on the one hand, including, for example, our diet, the air we breathe, and what is encoded within our genes. So this nature versus nurture debate has been an important factor in thinking about the evolution of many different diseases, of which cancer, I would argue, is perhaps the prime example. And this has really been the focus of my interests over the last 25 years or so. So to give a background to what I do, let me remind you about things that I'm sure that all of you appreciate. The idea is that we are made up of literally trillions of cells of many different types, and each of these cells carries within it what I call an instruction book, written in its own DNA. And of course, this instruction book is coded in units called genes, which are organized within the genome. Now, here's something that many of you may not appreciate. Even as you sit here listening to me talk, your DNA is changing. It's mutating because DNA is not chemically stable. And due to metabolism within cells and the effects of environment, that DNA is changing all the time. And this is clearly one of the key factors that is showing up as changes in DNA which accompany the evolution of cancer. As I'm sure all of you are aware, our ability to sequence DNA at large scale has grown pretty fast. This graph takes you only up to 2014, but it's continued its downward trend. We are not far away from $100 whole genomes. And this has really shown up first and foremost in cancer research, because, as you know, cancer is driven by changes in our genome. And we've gone very rapidly in the last 15 years from sequencing the first simple cancer genome all the way through to today, when thousands of cancer genomes have been sequenced, and this information is being used to treat patients and indeed to undertake tests for early detection of cancer in the blood. By the same token, the amount of information that we have to collate, curate, and analyze is growing rapidly. And it's gone from terabytes to petabytes to exabytes now, and it promises to grow even more significantly in coming years. This is really one of the major challenges facing us, how to fully unveil the potential of all the genomic information that is being collected about human diseases, particularly cancer. So as I'm sure you're well aware, gene mutations cause cancer. And what we know from the studies of the last 2 decades by a number of different laboratories is that as cancer progresses, shown here in the case of the colon, where you go from normal-looking intestinal epithelial cells all the way through to frank invasive carcinoma, the progression of cancer is accompanied by the gradual increase of alterations in DNA. And this process, called genome instability, is a universal feature of cancer cells. It makes these cells behave in ways that are abnormal, which in turn confers the behaviors that we collectively term as malignancy. As I mentioned to you a moment ago, at least in part, the drive for mutation is intrinsic. DNA is not stable and it is mutating even as we sit here, minute by minute. You expose, say, a centimeter square of your skin to the sun, and the ultraviolet radiation is sufficient to cause hundreds, if not thousands, of mutations. And all of this has to be combated in order for us to preserve genome integrity and suppress the onset of cancers. But genes are obviously not the only element in the origins of cancer. I'm sure you'll be well aware from well-documented historical examples like the onset of cancer of the skin in chimney sweeps in Victorian London because of their exposure to coal tar derivatives, which are mutagens in the chimney soot, all the way through to a more recent example of smoking, which causes lung cancer, again through environmental exposure to mutagenic chemicals. The environment plays a major role. So my work over the last 20 years or so has really been focused around two very simple but very complex questions. What is it that makes some of us more susceptible to cancer than others? And what is it that we can do to delay or prevent cancer onset in the majority of people? And to do this, what we have been doing in the lab is first to study the mechanism of high penetrance germline mutations that cause cancer susceptibility in order to try to answer this question. And on the other hand, we have been developing new integrated technology platforms that can pass the complex mechanisms involved in cancer susceptibility in order to develop new ways to treat, delay, or prevent cancer onset. So we know now, and this is data from the TCGA project at the NIH, that in certain forms of cancer, shown for you here at the bottom of this graph, people who develop these forms of cancer, they include canceres of the ovary, pancreas, and breast, for example, they actually inherit a very high number of these types of inherited gene mutations, which appear to make them more susceptible to cancer. And indeed, my own journey started some 15 or 20 years ago when it was discovered that a complex of genes, in particular these two genes, the so-called breast cancer genes, BRCA1 and BRCA2, and their associated gene, PALB2, partner and localizer of BRCA2, are mutated in the germline in many of these families. What we know now through the work of many different groups across the world, including my own, is that in the case of breast cancer, germline mutations account for only about 5% of all cases, and they involve mutations in high penetrance germline genes such as BRCA1 and BRCA2. And people who inherit these mutations have a very high risk indeed of developing cancer by the age of about 70 to 80 years. But what is also striking is that these cancers are early onset. They tend to occur earlier in life than is usual. We also know that many of these same faults actually occur in the 95% or so of cases that are sporadic. In other words, they have no obvious family bias. So what we learn from studying this route of cancer susceptibility applies also to the vast majority of cases. So how do BRCA2 mutations actually increase the risk of cancer? The idea was formulated in the so-called two-hit hypothesis for tumor suppression by Al Knudsen in the 1970s. And when we started this work, we thought that BRCA2 was a two-hit tumor suppressor, in that you would inherit one defective copy of BRCA2, and in certain cells, the second copy would also be lost, and this is what triggered cancer. Of course, we now know that this is not always the case in BRCA2 germline mutation carriers. And we also really do not understand this question, which is, what is it that makes these people who inherit one defective copy of BRCA2 more vulnerable to the second event, the loss of the second allele? So over the last couple of decades, my lab has gone from showing first that, or discovering the function of BRCA2 in maintaining the integrity of the genome, by showing that the inactivation of BRCA2 leads to aberrations, which I hope you can very clearly see in the structure and the number of chromosomes that encode or contain our genome. Over those 20 years, our journey has taken us from molecular cell biology and genetics all the way through to structural biology, super resolution microscopy. And this recent cryo-EM structure from my laboratory shows, for example, how certain segments of BRCA2 regulate the repair of DNA breakage by influencing the assembly of a complex enzyme called RAD51, which actually repairs the DNA. And we've also used analytical techniques, biophysical techniques, such as super resolution microscopy, to chart in great detail at molecular resolution how BRCA2 works to enable RAD51 to form these beautiful elongated filaments at the sites of DNA repair in cells. But does the environment play a role? So if you inherit one mutant copy of BRCA2, what is it that then makes you more vulnerable to carcinogenesis? And what we have very interestingly and recently been able to show is that there may be a chemical trigger for mutagenesis and carcinogenesis in carriers of BRCA2 mutations. As you may know, aldehydes are chemicals found not only in our environment, for example, in cosmetics, car exhaust fumes, in wood glue made by industrial processes. These are all over the place. But also, aldehydes are made by our body's own cells in very small quantities during normal cellular metabolism. And what we have recently been able to show in my lab is that in people who inherit one defective copy of BRCA2, aldehydes can induce the proteolysis of BRCA2 protein, and this in turn reduces the amount of BRCA2 protein below the threshold that is functional. Normal cells are therefore not affected in a similar way, and this leads in turn to genome instability. So we have this interesting clue that perhaps environment and genes, in the case of BRCA2, collude to cause cancer susceptibility. And this is a major and interesting avenue of research we are now pursuing in my lab. I can't help but say that it also led to perhaps our highest profile publication. For those of you who are in the UK, you will know that The Sun is a tabloid newspaper with a circulation far greater than Nature, Cell, or Science. It's in the millions. And of course, they ran this headline story upon publication of our paper, saying that booze, shampoo, and car fumes can all cause cancer. It's the first time I've been called a boffin. But our work has not only led us to understand how cancer originates in BRCA mutation carriers, it's also led to increasing insight into how we can treat the disease very effectively. And the simple graph taken from Nick Waddell's paper on pancreatic cancer published a few years ago shows you quite clearly that individuals here who inherit BRCA mutations actually respond extremely well to a subset of cancer drugs. So these patients can now be treated very effectively. And someone whom I trained as a medical student in Cambridge, Serena Nick Zainal, her lab has very recently shown that you can develop a test for a biomarker that can predict this responsiveness. So we've come a long way in 20 years from understanding that BRCA2 mutations cause cancer to understanding the molecular mechanism all the way through to offering very effective treatment options for patients who suffer from this form of the disease. But there are major new horizons still to cross. And in our lab, we've been developing a series of new platforms to accelerate the discovery of next generation medicines. These range from new genetics tools which can pass complex biological pathways and directly identify and validate new therapeutic targets, to the combination of structural biology with chemistry to enable structure-guided lead discovery as a starting point for new drugs. And with Alessandro Esposito's lab, we've been developing new microscopes which integrate the capability to interrogate all aspects of photophysics in a single instrument in order to reveal how new drugs actually work within cells. So let me simply end by saying our journey is far from over. A lot of this work is being spun out and translated in industry, for example, in Foremost, which was a spin-out company from my laboratory. But there are many new horizons, and I would just like to end with something that one of my own mentors in Cambridge shared with me many years ago, and which I'd like to share with you. And this is paraphrased from the French philosopher André Gide. And it simply says, "You cannot discover new lands until you first consent to lose sight of the shore." I hope that many of you in the audience will join us at the Cancer Science Institute of Singapore to undertake new voyages in your research. Thanks for your attention.
Thank you very much, Professor Ashok. Now the floor is open to all the attendees for questions.
I see no open questions. Perhaps people are using the chat function, so I'm going to
the question, no, it's not. Only the question and answer. Maybe I'll just ask a very simple question. So what advice would you give to people who are considering to do PhD? What kind of research should they go into? Because cancer has been, we have been dealing with cancer for a long time, yet we have a lot of breakthroughs, but we have not cured cancer yet. So what kind of advice would you give a PhD student? Right.
That's a very interesting question, Caroline. And the way I will put it is this. I think we are nearing what I call the end of the beginning in cancer research. So we have a good understanding of what genetic alterations cause cancer.
Mm-hmm.
We are beginning to understand which of these genetic alterations occur in different forms of cancer arising in different tissues. So I think the stage is now set for us to be able to stratify on the basis of alterations in the genome, different types of cancer, and begin to develop, as I alluded to in my last few slides, new ways to develop next generation medicines against these forms of cancer.
Thank you.
So I think that is the big question. So I can see some questions coming up, and I'll take them one by one.
Yep.
So the first question is, how can a medical student gain experience and research skills at CSI during this COVID outbreak right now? So the COVID outbreak has definitely impeded, if you will, the ability for people to interact with patients. But as Singapore moves towards a gradual lessening of internal restrictions, research at the CSI in the laboratory is very much on the go, nearly back to full capacity. So I don't think that the COVID outbreak will affect the ability of medical students to gain experience of research. How interdisciplinary is the field of cancer? It's always been interdisciplinary, and Mohammed, who has asked this question, I hope you can see from the very short presentation that I gave, it's extraordinarily multidisciplinary, at least the work in our lab. We use the techniques and methods of molecular cell biology, including super resolution and dynamic imaging, all the way through to structural biology using both cryo-EM and X-ray crystallography. And we also use many chemical and biophysical tools in analyzing pathway effects and trying to discover ways to discover or create new drugs. There's a question coming up asking, do I accept students in my lab? Yes, of course, I'm interested. Does one have to have experience in cancer research in order to come to my lab? All I can say is I started my life as an immunologist studying the immune system, and I moved to cancer research because I got interested from trying to understand how genes reorganize during immune system development to understanding genomic instability in cancer. So no, I think what you need is not experience in cancer research, but relevant experience, the ability to think through problems. And you need to be able, I think, to demonstrate your analytic capability and your ability to learn new experimental techniques rather than necessarily to have prior experience. Runze is asking, is there a PhD opening in the Research Institute, and what will be the required background and application materials? Yes, there are openings, not just in my own laboratory at the CSI, but all across. Please visit the CSI e-booth, which is open now. I will be coming there for a live chat in a little while. But you will find, I think, all the answers that you require regarding the openings in the different laboratories in the Institute and how one goes about applying to these.
Okay, any more other questions? You can even raise your hands if you like to. Okay, there's not many questions. Professor Ashok, would you like to have some last words?
Yes, of course. I think really,
There's one more
what are
question.
some of the current problems that you are working on? Really, our focus is in a few areas. The current problems that we are working on are, first, how do mutations affecting BRCA1, BRCA2, and other high penetrance genes perturb cellular functions in order to cause cancer? So we understand that BRCA2 is involved in genome repair by homologous DNA recombination, but we have also very recently discovered functions in the regulation of transcription and the regulation of chromatin structure. So we are working on the idea that some of these new functions of BRCA2 may also lead to genome instability or in other ways promote the genesis of cancer. The second major focus within the laboratory is to address this issue of susceptibility via aldehydes. Now, it's not just BRCA2 deficient cells that are affected by aldehydes. As you may be aware, aldehydes are mutagens that are present, as I said, in the environment, but also are high in many different conditions, for example, metabolic syndromes. Is this what leads to the increase in cancer incidence in patients with metabolic syndromes? This is an important issue that we're working on, combining genetics, molecular cell biology, and somatic cell genetics. And finally, as I said, the new platforms that we have devised have led us to be able to unlock many different new cancer susceptibility pathways. We've identified some very interesting new molecular targets, and we're on course to try to develop new drugs to be able to see if we can delay, prevent, or treat cancer. So due to the COVID pandemic, many campuses are closed. Is a virtual lab useful and valuable experience? Yes and no. In some disciplines, it is certainly very useful, but these would primarily be what I call the quantitative disciplines. Unfortunately, much of what we do still involves experimental wet bench research, so it's not possible to fully recapitulate that in a virtual lab. Do I think that wound healing and regeneration could give us some insight into therapeutics for cancer? That's a very interesting question. And the short answer is yes. Wound healing and regeneration, if you will, are normal processes which are self-limited. And in a way, cancer could be a manifestation of similar processes in which the normal limits are lost. So I think studying this would be a very fertile avenue to get insights into the abnormalities in cancer.
Okay.
What motivated me to pursue a PhD in immunology or cancer research? If I'm honest, after I finished training as a physician, I did my medical degree, I was working in the wards, and I was increasingly dissatisfied with just doing things rather than understanding why I was doing things in the clinic. So I got interested in research. Why did I go into immunology? Quite simply because I encountered, when I was working in the hospital, a number of infectious diseases which interested me. And I thought that understanding how the immune system combats infectious disease might be an interesting way. So in effect, I entered research because of my own motivation to answer questions which were arising all about me. I certainly did not have any long-term career trajectory in mind. I've simply danced to the beat of my own drummer, I think, throughout my research career. Why did I change to cancer? Well, I was studying VDJ recombination or genome rearrangement in the immune system when I started my own lab up here at the MRC in Cambridge. And we were getting interesting insights into how the break repair process in DNA is regulated in the immune system. And that motivated me to ask the question, what happens then? How is control of this process lost in solid tumors where you get many structural breaks in chromosomes? And I was lucky because at the time, BRCA1 and BRCA2 had just been identified. That prompted my lab to immediately create new models for BRCA2 deficiency, and we found that it actually controls genome stability. And that discovery has really motivated the last 20 years of my research. So thank you all for listening to me, and I hope that many of you will be interested in applying to the CSI, not just to my own lab. Please visit the CSI e-booth. Thank you very much, Caroline, for hosting
Okay.
this.
Thank you.